Okay, good afternoon, everyone, and welcome back from your spring break. I hope that you managed to catch up on everything you needed to catch up, which includes sleep. Today we're going to be carrying on discussing a bit of physical evidence, and that is hairs and fibers. Hairs and fibers are very, very, very frequently found at the scenes of a crime. And they offer up tantalizing possibilities of useful evidence. It used to, I have to tell you that it used to be that hairs and fibers were accorded an enormous amount of significance. But that has changed in more recent times. And the weight of evidence of hairs and fibers is now somewhat diminished for reasons that I'll point out as we go along. However, hair at least is enjoying something of a revival of interest because hair very often carries DNA with it. Hair is a cellular material and a cellular product, and it may have DNA associated with it. So I'll explain more about that as we go. We're going to be hairs being a biological product, fibers being an artificial product, a synthetic product. That will be our distinction. And I'll have rather more to say about hair than I will about fibers. So hair is, the possession of hair is a characteristic of all mammals, except for some of us. But even I produce hair because I am a mammal. It's a characteristic of the mammals that they have hair. And one of the features of hair is that an individual hair has only a set lifetime. And it is maintained in the hair follicle for a period of time. It ages and dies. The root dies and the hair then falls out. So we shed hair continuously. And you'll know this is a completely natural part of the process, that we shed hair on a continual basis. So the presence of hair at any scene is no surprise. Anywhere you go, when you do your housekeeping and you vacuum, you look at your vacuum cleaner and you'll see that it is absolutely full of hair, especially if you have pets, for example. But if you look at what's the contents of the vacuum cleaner, you'll see that it contains an abundant human hair as well, if there are people living on the premises. So hair is going to be encountered at virtually every crime scene. If there is no hair present, it's a strong indication that that crime scene has probably been cleaned up and vacuumed to remove evidence. So it's very tantalizing that we have hair present at crime scenes, and it would be wonderful if we could just look at the hair and be able to recognize individual characteristics in that hair. But unfortunately, that is not the case. At best, hair on its own usually presents only class characteristic. It does not possess individual characteristic. It used to be thought that an expert could distinguish individual characteristics in which could tie questioned hair to sample hair. But it turns out, under scientific investigation of that process, that this is subject to a tremendous amount of error. So that is why hair evidence has lost a lot of its significance in more recent times. So hair has a morphology, it has a structure, and the morphology of hair does vary amongst groups of people, and also, of course, on the different places on the body where that hair grows. It may have different characteristics as well. Hair can, although the individual characteristics of hair are somewhat limited, it can provide strong corroborative evidence under certain circumstances. If hair recovered at the scene is anomalous to that scene, and yet can be successfully compared to hair from a suspect, for example, then it provides corroborative evidence of an association, but it is not definitive. So again, what are we doing on all of these things? We are doing pattern analysis, right? We are comparing patterns that are present on hair, on a questioned hair. We're going to compare those to hopefully a standard sample from a suspect's residence or whatever. So we'll have a look at the structure of the hair and see how these characteristics arise. So first of all, hair is a product of the skin, and it is produced from special cells which lie in the dermis, in the deep parts of the skin. These cells are actually very interesting cells in biological terms. They're actually stem cells, and they form a little bud of generative cells, which generate very rapidly, generate specialized cells which pack together to form the hair. So hair is actually made up of thousands. An individual hair is made up of thousands of cells which have been specially processed by the body. They are richly invested with a particular protein called keratin. It's the same material, same protein that makes up our fingernails. And these cells are richly packed with keratin, and they are packed together. They are no longer living. Very shortly after the production here, they actually cease any metabolism, get packed together tightly, bound together by their keratin to form this very tough structure, which is the hair. Hair is actually very strong. A single hair can lift, if you do it properly, can actually lift more than a kilo in weight. They're actually strong. Human beings have relatively few hair, okay, compared to other mammals. We are rightly referred to as being the naked apes because we actually don't have a lot of hair, but nonetheless, we do have hair, and we continually produce hair, and we continually shed it. So it may have some particular significance. So that little generative bud of cells there is housed in part of here's the dermis here, extending all the way down into a follicle. It's an enclosure, an envelope, which roots the hair. So this we refer to as the hair root. When the hair is alive and growing, it is tightly bound to the root there. It can be removed if you pull on it violently. Even a living hair can be removed. When you do that, it's important to note for forensic purposes, you may carry with it a portion of the living tissue that normally surrounds the hair. So here's the structure of the hair itself. It's actually quite complex. It's not just a thread. In the center here, it has packed proteins that form like rods, and that's called the medulla. Now, the structure of the medulla varies a lot. And this is one of the things which a hair expert will examine, will look for as a characteristic of the hair. Sometimes, in some people, the medulla is almost entirely absent. It's just a hollow or collapsed tube in the center. In others, it's packed tightly with protein. In others, as we'll see, the protein rods may be broken up. So you may have a bit of protein, then a space, bit of protein, then a space. So the medulla can actually have important characteristic structure to it. It may also be pigmented or non-pigmented. So these are other characteristics. Around the medulla, there's a cylinder here called the cortex, and the cortex is where the pigments of the of some of the pigments of the hair lie. And if you look carefully, you'll see that here you can actually see the cell shapes. The cells are no longer alive. They have been preserved in this flattened structure, forming this dense cylinder around the medulla. They may still have DNA material in them. So this is a possibility. It's not always a good possibility, but it is a possibility that you can actually extract DNA of a particular kind from these cortical cells. So they form this dense cylinder, which is the major body of the hair. And then around the outside, there are flattened cells, which almost like shingles on a roof, laid down one over the other. And this is the hair cuticle. And it has these like scales. The cells have become so flattened that they form scales around the outside. And this is the cuticle of the hair. And that is the structure. And each of these portions of the hair may have characteristics which an expert might be able to describe. So first of all, the cuticle itself, becomes important because there are many, many kinds of hair present virtually everywhere. All mammals have hair. And all mammals have this characteristic of the hair that it is continually shed. It's continually produced and continually shed. So in any premises, at any crime scene, whatever, you are likely to find hair of many different kinds. In addition, hair is very light and it can blow. It can be blown into a scene or carried in very, very easily. The very first thing that an expert has to do is present it with a hair sample. They have to decide whether or not that hair sample is actually human hair or not. And the one really clear distinguishing feature is the structure of those scales that make up the cuticle on the outside. These here are non-human, things like dog, rat, a cat, these sorts of things. They have very clear scale structures to the cuticle. This is the human hair. And this, you see that it has these irregular, very flattened scales. This is called an imbricate pattern. I-M-B-R-I-C-A-T-E. I-M-B-R-I-C-A-T-E. Imbricate pattern. And this is characteristic for human hair, no matter where on the body the human hair comes from. There is one other thing just to point out. The scales here are laid down one on top of the other, and they point towards the tip of the hair. So the tip of the hair would be here. Since these scales are laid down like that, the tip of the hair is here. The root of the hair would be down here. So here is the typical human imbricate flattened scale type. And it's typically with these irregular edges, and these scales do not project the way that animal hair very often does. Here are some other ones for you to see. This is human hair. Again, this is typical imbricate. And where is the tip of the hair? It's down here somewhere. Here is dog. This is dog hair here. This is deer, rabbit, cat, and this is horse. Now, some of these may look similar to one another and have imbricate pattern as well, but an expert can quickly distinguish which of these is actually human hair and which is animal. That is not to say that animal hair is not important. Animal hair may be important. because you may be able to, for example, if a victim has cats and the suspect does not have cats, and yet clothing that a suspect has worn and is recovered from their home has cat hair on it, which matches the cat hair from a victim's, from a crime scene, that is a strong implication. It's not proof, but it is a strong implication that there is an association, that the suspect was at the crime scene. And animal hair is very, very easily picked up. It's very, very easily acquired. It's often very abundant, as anybody who has cats or hairy, furry dogs knows. It can be very abundant. So here's the one of the most important characteristics of hair, and that is its color. Most of the color, most of the pigmentation of the hair is lodged in the cortex. Now, I'll remind you, this is the cortex. That's the main body of the hair, the cylinder of cells, which are not completely flattened the way that the cuticle is. Very, very tough, thick compared to the rest of the hair structure. And it's the pigment exists as granules within the hair. The color, the shape, the distribution of the granules can provide some important information. However, it transpired that, in fact, there is a considerable amount of subjective decision made about color and about its relevance, and how similar, for example, one color is to another, how similar a questioned hair sample color is to standard, to hair collected under standard conditions. So it is important, but again, it has to be approached judiciously, because it's not going to provide individual characteristic. It's going to provide class characteristic. You just see here that there actually are sometimes very obvious differences. Blonde hair, for example, which has very little pigment compared to dark hair, brown, like brown or whatever. A wide range of hairs needs to be collected, because hair differs tremendously in its coloring. Even on the same head of hair, hair may vary in color from one region to another. For example, even young people have gray, often have some gray hair which has lost its pigmentation entirely. So first of all, at a crime scene, a wide range of hairs needs to be collected, if that is at all possible. And under standard conditions, a wide range of hairs needs to be collected for the comparison to be effected. So the medulla provides another element of characteristic. That is the center portion here. And here you can see here, it's very, in humans, extremely thin. And it may or may not be pigmented. It's another characteristic. In this case here, the entire medulla here is deeply pigmented. In animals, it's often very much larger, the medulla. In humans have the characteristic, it's usually very, very thin. And in fact, in some instances may be almost entirely absent and have almost like a collapsed look to it. That the proteins within the medulla can be continuous, as you'll see here, that is continuous. But they may also be interrupted, almost like, what would you say, hyphenated in a way. If they are widely spaced small portions, which are very widely spaced. It's referred to as being fragmented. And sometimes the medulla may be entirely absent, as seen here. So this is continuous. Here is interrupted or fragmented, depending on how wide the spacing is. And it may vary, by the way, in different parts of the hair. And this is the medulla is entirely absent. So again, here are sets of characteristics, but again, they are class characteristics. They are not individual characteristics. So there's a little bit more of the structure of the hair root and everything. Here's the hair follicle. This is dermal cells, and here are the generative cells of the root here, which are generating all of the cells which get packed together to form the hair itself. And this distinct bulb that forms around the generative cells also has a blood supply. This is called the papilla. So this is the hair papilla, and if you actively pull the hair out, you can see it quite easily with the naked eye. You can see the bulb shape at the bottom of the hair root. Now, just a couple of other things about the structure of the hair. Here's the hair root here, but there are two things associated with a hair follicle. The first is hairs have muscles attached to them, a very, very small muscle. Here it is here. It's called the hair erector. And when it contracts, it pulls the hair up, pulls it upright. We've all had that experience, okay? If you get a fright, or if you are emotionally moved or sexually aroused even, the hairs on your body, often in specific areas of your body, you will feel them rising. You'll feel them standing. And made my hair stand on end is literally true. The hair does stand on end, and it stands on end because of this little muscle here pulling on the hair, pull it upright. It's a hangover of our evolution. Many animals who are densely covered with hair or fur, when they are alarmed or need to fight or display in some way, will erect the hair, all the hair. You've seen it on a cat. Give a cat a fright, and what you see its tail puff up, and the whole of its body, all of the hair erects to make the animal look bigger, to make it more imposing and less likely to be attacked. In our case, you can hardly notice it. We can feel it. We can feel the hair standing up, but you don't really notice it happening to somebody else. Also associated with the hair is this. It's called the sebaceous gland. It's an oil gland. It secretes lipids, oils. These lubricate the hair, but they also lubricate the skin, and they also have an antibacterial action. In forensic science, of course, they are very important because this is one of the contributors to the fingerprint, to what leaves a latent print. The friction ridges of your fingers acquire oils from your skin and can deposit them when you touch a surface and to leave a latent print. I think that's all I need to tell you about the structure. So the hair has various growth stages. For much of its life, most of its life, it's actually in an actively growing phase. It just grows continuously, and you know this. We have to, unless you've grown your hair very, very long, lucky you, if you don't grow your hair long, you have to continually cut your hair. And that is because the hair actually lives for about 6 years in a human being. And it just grows continuously. And that is for most of our body hair. We have a type of hair which does not grow so actively and forms a soft, downy hair, which is almost invisible. Very often replaces hair. Once hair falls out, it may get replaced by one of these downy hairs. But The majority of the hair that we have grows continuously, and it is continuously in just this little papilla at the bottom, and that little bud of generative cells continually produce the cells, and it's like a conveyor belt. It just proceeds out of the follicle continuously. That whole stage of active hair growth, when the root is alive and producing hair, is called anagen. And as I say, it lasts up to 6 years. But then after about 6 years, the growth of the hair slows down. It slows down, and if you look, you can see that the follicle is maintained, but the follicle kind of withers. It becomes much less active. And this is called catagen. And the hair is still alive, it is still held, but its growth has substantially stopped. Then the hair follicle actually ceases activity, and the root itself dies, the growth stops, and the hair loses contact with the follicle and becomes loose and is easily shed from the follicle. It can be pulled out easily, or else ultimately it just falls out. So those are the 3 stages: anagen, catagen, and telogen. And hair which is collected as a sample should be classified into those 3 stages. You may, at the scene of a crime, the presence of abundant anagen and catagen hair may be an indication that hair was actively pulled out. Hair can be pulled out during a struggle, not necessarily by people grabbing it and pulling it, but can be pulled out just through friction, through contact with something else. And hair which is free at the scene of a crime, which is patently in anagen or catagen, it's symptomatic of hair that has been pulled out. Telogen hair is hair which falls out easily and might well have been shed beforehand. It may be present at the scene just naturally as part of the fact that people were around shedding their hair. So here are the different stages. This is anagen, and you can recognize it because here at the bottom, there is a distinct bulb here. And if you were to slice this open, you would find that actually there's a little papilla in there that is producing the hair itself. Around this, by the way, this has been pulled out, and around it there is tissue here. But the hair is living and is still actively growing, and is growing from this portion here. This is catagen. This is hair which you can see how much smaller that this is compared to this. And it may still be alive. It may still be still be maintaining living cells at the bottom here, but the hair is no longer growing. But it is still rooted into the skin. This is a hair in telogen, and this is just the end of the hair. The root has died completely, and all contact with the root has been lost. And instead, this hair has no living cells in it, and it's ready to be shed. Now, if you take a living hair that is in anagen here, and you pull it vigorously, you can easily pull it out. The hairs are not that deeply rooted into the skin. You can easily pull them out with enough force. And I'm sure that you've probably done this at some stage in your life, either deliberately or accidentally. And if you look at that, there is something which is potentially of enormous forensic value. When you pull out a living hair, it carries with it a tag. The whole thing here is called a tag. Here's the root at the bottom. This is in anagen. So this is a living root. The hair cells at the bottom here are alive and tightly tied to the root, to the follicle. And when you pull it out, you pull out the entire follicle with living tissue on either side. The reason that this is so significant is that this is a very rich source of DNA. So here is a chance. All of our other characteristics that we've talked about with hair give us only class characteristic. But here is a chance for hair to provide us with individual characteristics, because this is a rich source of DNA. If hair like this is recovered, which it quite often is, for instance, if there has been a struggle and a victim has grabbed the hair of an assailant, they may pull out hair from follicles, and that can provide us with DNA, with a DNA sample. That's not the only source of DNA in hair, as I'll explain in a minute, but it is a very rich source, very important source. So any hair seen at the scene which has this characteristic, has characteristic skin tag associated, follicular tag rather, associated with it, is very carefully collected. So apart from the possibility that hair may carry DNA with it, the hair expert has to be able to compare hairs. Again, it's another of our pattern recognition systems. We have the hair sample is useless just on its own unless we can compare a question sample to a sample collected under standard conditions. And here again, we can make use of this tool, the comparison microscope, to try and compare questions with standard sample. And the three things that we've discussed are particularly important. First of all, the outer scale structures. Second of all, the size of the medulla and the medulla shape. The size is called the Medullary Index, and it's a measure of how wide the medulla is compared to the rest of the hair. The medullary shape is what we talked about before, whether it's continuous or in that hyphenated state, or fragmented or absent. So, and the hairs, their width, for example, can be compared, but with a lot of judgment being involved, because hair can vary in width. Even on your head, different hairs may have different widths. So if the expert is going to compare hair, they usually compare as big a sample as possible to get a range of variability to effect the comparison. So again, we're going to look particularly at color, the length, diameter, all of those kind of things, structure of the medulla, the color of the pigment granules, which are present in the cortex. All of these things used to be thought to be very close to individual. And this kind of hair evidence in some cases proved to be extremely, extremely important, if not definitive in some cases. However, subsequent investigation by the FBI has found that there is a tremendous amount of error involved. So I'll just explain to you that when an expert comes in to a court of law, that expert is testifying based on their expertise. Their findings may well be challenged by a defense expert who gets a chance to examine exactly the same piece of evidence. But here is the problem. It's just a human problem. And that is that these two experts are on two different sides. They are not necessarily being dishonest. They may well be testifying according to their ability, but in scientific terms, we have very little way of assessing who is correct. We have little way of determining if this person is right, how right are they? If this person is wrong, how wrong are they? And for that reason, the FBI, for example, has conducted scientific experiments where they have taken samples, which they have deliberately manipulated. They've taken question samples and then a standard sample from the same source, or a supposed question sample, and a standard sample from a radically different source, etc. They've set up all sorts of different situations of comparison and then asked a variety of experts to assess these. And that is a proper scientific experiment. And unfortunately, what happened was that had a very high error rate. Even the experts could not, in many cases, come to agreement on what was correctly associated and what was incorrectly associated. Which of these samples, question samples, had strong association correctly? Which of these samples had no association with the standard samples correctly? There was too much error in the expert analysis. And this was a tremendous blow to the science of hair analysis. The amount of error was substantial, 11% in actual fact. Came up with erroneous findings. And we need to realize that is more than 10%, more than 1 in 10 trials could have incorrectly sent somebody to prison for life. In other words, based on this evidence, it's far too high to be acceptable. So nowadays, hair analysis is much more likely just to be regarded as being corroborative, and an expert will state all the caveats. And the experts, by the way, now are, I would think, a lot more humble about their analysis and about what they can actually say in hair comparison. If the only chance really of a positive ID being gathered from hair, from a hair sample, okay, is if there is DNA present associated with the hair. There are two kinds of DNA, by the way, which can provide evidence. The first type of DNA is nuclear DNA. I'll show you here. If I can get back to this. Do you see here in the cortex, you see that there little dark dot? That is the remains of the nucleus of the hair. And it is possible that there may be DNA, enough nuclear DNA in there for us to do an analysis. The problem with this is DNA decays very, very quickly. So the possibility of extracting DNA sample from hair itself, nuclear DNA, it's called nuclear because this is the nucleus. So the possibility of extracting nuclear DNA from a hair is actually quite remote. The nuclear DNA decays very quickly. However, there's another kind of DNA which lasts much, much longer. And this is mitochondrial DNA. So there is a spot for you there. The mitochondria are little organelles inside our cells which produce energy. So as long as that cells were living down at the base of the hair, they had active mitochondria. But the mitochondria have their own DNA. And you cannot individuate mitochondrial DNA. You share your mitochondrial DNA with all of your siblings, and you inherit your mitochondrial DNA from your mother. Your mother inherited it from her mother. So in other words, you share your mitochondrial DNA with all your maternal cousins all of those relatives, all of those cousins of yours. A cousin is somebody with whom you share a grandparent of whatever status of grandparent, great-grandparent, great-great-great-great-grandparent, great-great-great-great-grandparent. Your maternal great-great-great grandmothers all handed on this same DNA. You share that mitochondrial DNA with all the cousins of your maternal line. So mitochondrial DNA doesn't allow us to recognize an individual, but it is very strongly corroborative. If a suspect has the same sequence of mitochondrial DNA as a question's sample, it is very, very strongly suggestive that you're dealing with somebody closely associated with that crime. We will talk more about this kind of DNA association, this DNA analysis, when we discuss DNA. So for the moment, just realize that hair offers us two possibilities as a source of DNA. The first source, if we're lucky, the hair has been pulled out while it was still alive, and it carries with it tissue with living cells, which are a rich source of DNA, which can be analyzed to provide an individual identification. That's possibility one. Possibility two, if we're lucky, there might be some DNA, nuclear DNA, in the hair itself. It's very unlikely, but it is a possibility that can be tried for. But much more likely is that hair, even aged hair, will contain mitochondrial DNA, which may point us in the right direction. Won't give us an individual characterization, but it will at least point us in the right direction. We'll know it's like a narrow class of evidence. Okay, so as I said, this, apart from the DNA analysis, which is very accurate and can be, it's truly scientific, its errors can be very carefully determined. The rest of hair analysis has fallen under something of a cloud. So there's just some questions about hair. First of all, can the area of the body that hair came from, can that be determined? Yes, it can, within limits. That depending on where the hair grows, head, armpit, beard, body, pubic hair, eyebrows, eyelashes, all of these hairs do have some characteristics, but their characteristics sometimes overlap. So you can get beard hair, for example, which looks remarkably like pubic hair. You can get short head hairs, which look like eyebrow hair, etc., etc. So it's, again, an expert will ask, try to obtain a wide sampling in order to effect an accurate comparison. What about determining race? Yes, within limits. Ethnic characteristics, racial characteristics are imparted to hair, but you can see a grouping like European, Caucasian, African origin, Mongolian, etc., but there is tremendous overlap. So again, an expert will sometimes be able to say, yes, this definitively did come from somebody with African ancestry, okay? But very often the expert will say, well, it's likely, or it appears likely that this came from, but we'll have those caveats because there is overlap between the different groups. It's not always definitive. Age and sex, it's very difficult. It's very, very difficult to determine sex from hair. Likewise for age, infants have particular characteristics to their hair before their mature hair grows. So an infant's hair, the age can be determined. But once mature hair begins to grow, it's extremely difficult to tell any of those characteristics from it. Was hair actively pulled out? Yes, we've already discussed that. Very often, if it's actively pulled out with violence, then it will carry a follicular tag. But it has to be said that, in fact, not much force needs to be used. You don't have to do it with great violence. You can do it quite easily. And you can even remove hair. If you've got long hair and you're brushing your hair and the brush gets a little bit caught and you pull, it's likely that you'll pull some hair out, especially hair which is in catagen, for example. But this may be very important because, for instance, if we want to actually get a standard sample from somebody maybe somebody who's missing something like that, a hairbrush, for example, is a very good source of hair because it may well have pulled hair out with skin tags, which will allow us to make a DNA identification. When somebody dies, the hair follicle dies. The skin dies relatively slowly compared to the rest of the body. It will last some time in tens of minutes to hours. That sort of length of time. The cells could be formally said to be alive. They're not receiving oxygen, however, and they very soon start to exhibit all sorts of characteristics of death and decay. One little myth: your hair does not continue to grow after you die. Your hair, the cells in that little papilla at the bottom, they die very, very quickly, and hair growth stops almost immediately. So it is a myth to say that hair grows after death. It does not. There's two reasons why people think it is. The one is, after death, the surface of the skin may dehydrate and thin and pull away from hair that is already present in the follicles. So it appears as if the hair is growing. But the other is much more important, and that is that skin often pales markedly with death, and the dark hair becomes much more prominent. So these are things that led people to think that hair carried on growing. It does not. It stops growing very, very quickly. And in fact, the whole structure of the follicle changes. And you can see here, it develops this strange clubbed look to it. There is a point in the decomposition process where hair is very easily shed from the decaying corpse and actually falls out, and it will often fall. That hair will often have these characteristic clubbed ends with this darkened banding at the bottom. Okay, so I think I've explained this to you. The follicular tag can be a rich source of DNA because that follicular tag is usually hair, which is in anagen, as opposed to catagen or telogen. Hair in catagens can still provide DNA. Hair in telogen is unlikely to be a rich source of DNA. So hair, ordinary hair which has been shed already lying around a scene, very unlikely to provide nuclear DNA, but might have some mitochondrial DNA. Okay, I think I've explained all of this. So in general, this hair is very present, is carefully collected. If there is too much of it, it may even be vacuumed, but a wide range of samples of hair will be collected from the scene. But remember, those have to be compared. And the standard sampling of hair involves taking at least 25 full-length hairs from the skull, from various places in the scalp, because remember that even on your scalp, hair structure may vary. In cases where there has been a sexual contact of any sort, then pubic hairs will be collected as well. And pubic hairs might be collected from a range of suspects. And in addition, might even be collected from partners who are not suspects, because we acquire hair from other people that we live with, even not necessarily even during sex, although that's a rich source of hair. But we acquire hair from the people that we live with, on our clothes, on our bodies, trapped in our own hair. An investigator might see an anomalous hair, for example, and collect it. And then that needs to be compared to everybody to determine, is this from somebody who's suspect, or is this from an innocuous source? The hair is also collected, by the way, at autopsy. And part of an autopsy will usually involve examining the hair very closely for anomalous hairs and fibers which might be present. And the issue of collecting pubic hair becomes extremely important in cases of rape. And in those cases, hair combings may be done to try and pull out all associated loose hairs, some of which may be from a perpetrator. Okay, so that's all we're going to talk about for hair for the moment. But we're going to also talk just very briefly about fibers. Fibers being synthetic threads. Synthetic threads are extremely common, but don't forget that we also have natural fibers. So, for example, we commonly wear clothing made of plant fiber, cotton, for example, and animal fiber as well, wool, that sort of thing. So all of our clothes, like a woolen sweater, for example, is likely to shed fiber. And those fibers might, in fact, be significant. And what an investigator will often look for is anomalous fiber. Okay, a fiber of a different color, a fiber which appears to be of a radically different structure to materials that the investigator sees in the surrounding at a crime scene, for example, may be significant. They're natural fibers, but there are many man-made fibers as well. And these are so numerous. There are so many different kinds that it is absolutely perplexing to try and classify them all. There are many different kinds, and every manufacturer has their own patented methods for producing that particular fiber. The first lot are referred to as regenerated fibers, and the examples of these are rayon, acetate, triacetate, a typical process for producing these. The rayon process, for example, is typical of this. Rayon is composed of cellulose. It's actually, they take natural cellulose of some sort, and they dissolve it. And they dissolve it, and then they reprecipitate it. The reason they do that is natural cotton, which is just cellulose fibers, varies tremendously in width and in length. And it makes it very difficult, and it makes it difficult to manufacture with constant quality. But regenerated fibers can use a natural source of cellulose, dissolve it, reprecipitate it, and then spin it into enormously long lengths of a constant width and a constant quality. So that's the huge advantage of using that kind of regenerated fiber. The example I'll give you, bamboo, you'll see these bamboo textiles, say bamboo, this bamboo, that. Bamboo textiles are actually rayon. They're a regenerated fiber. The source of the cellulose is bamboo. Then there are a whole lot of fibers which are entirely synthetic. They're effectively plastics. And largely derived from carbon fuels and fossil fuels. And these are all of the things like nylon, polyester, acrylics, there's just hundreds of them, of these entirely synthetic fibers. And they are polymers. What they do is they manufacture sets of specific molecules of a particular structure, and then they join them all together. And they join them all together into a polymer. So they are made up of monomers, an individual molecule, which is synthetic, and then they are polymerized. They're joined together and spun out into enormously long fibers for whatever purpose, which can be woven, turned into textiles, etc., etc., etc. So here is a huge difficulty, and that is that these fibers are unbelievably common. Our homes, our vehicles, our workplaces, everywhere, there are just thousands of sources of these synthetic fibers, the curtains, the carpeting, the upholstery, etc., 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 clothing. All of these this huge variety of different kinds of synthetic fibers all can contribute fibers to a scene. So it's the fiber analysis is an extremely difficult process, because you may have, first of all, you may have a few fibers which appear anomalous, but you take those fibers, those few fibers, and because there's such a huge range of different kinds, it's extremely difficult to say what those fibers are. It's extremely difficult to be able to identify those fibers, given the huge number of different fibers, the huge number of different manufacturers that there are. If an examiner is asked to examine something that has been made out of the fibers, a textile that has been torn, or a textile that has been cut with tools, with individual characteristics, then they may be able to say, okay, this piece of textile is positively identified as coming from, being associated with its matching edges. But the examination of synthetic fibers themselves is extremely difficult. I'll give you a couple of the characteristics of the fibers, but again, fiber analysis used to be somewhat less complicated, because synthetic fibers, there were relatively few of them. Nowadays, there are so many of them. Their presence is ubiquitous, and it's extremely difficult to link fibers found at a scene with fibers found elsewhere. It almost, by definition, can only provide us with class evidence, an association between fibers at a scene and fibers recovered from a suspect or whatever, for example, under standard conditions, very often will provide us only with class evidence. The first thing to look at is the actual structure and the color of the fibers. And this will be done usually microscopically, and can use a comparison microscope as well. Think back, I remember I described to you this refractive index of glass. These plastic fibers have very often a refractive index can be determined, unless they're completely opaque. A refractive index can be determined from them. a similar for refractive index between questioned and a standard may suggest an association. But here's the other thing. Remember, I told you that there is one of the complicating factors is that there's just a gazillion different kinds of fiber. Well, if you can examine a questioned fiber and a source of fiber collected under standard conditions, and they have a very similar chemical composition, it is strongly suggestive that they may be associated with one another because of the fact that there are so many different kinds. An association like that acquires value because so many other associations were possible. It's still not individual. It's still class evidence, but is strongly corroborative. If you can take a fiber and you could show that it has a very, very similar chemical composition to fibers collected under standard conditions, then an association might be made. Here are some of the other characteristics. First of all, fibers vary in shape. That shape is determined by the manufacturer. These are usually produced by melting a source and then spinning it out and allowing it to solidify, like squirting it through a nozzle, basically. And in that process, first of all, the cross-sectional shape is imparted. That is determined by the manufacturer. And in addition, the fiber may acquire striations. It might acquire patterning. And the fibers may be treated after they have been produced. That is this complicated sense of the presence of delustering particles to reduce shine. It's just one of the treatments that they do to these fibers to make it look better. But it may impart some characteristics to the fiber that can be recognized under the microscope. Here's a very famous fiber. And this is a scanning electron micrograph of a nylon fiber. Here it is here, there. It has this flat triangular shape with a concave underneath. And this fiber also had a particular color. It was a reddish color. And this similar fibers were recovered from murder victim in the famous Atlanta child murders case. And a match was made between a fiber recovered from a victim and carpets in the home of Wayne Williams, who was arrested and convicted for these murders. And this fiber, it turned out, was manufactured in relatively small quantities, and even smaller quantities were put into these carpets, which they had acquired. His parents had bought some years before. I have to tell you that this fiber evidence was enormously important. It was one of the very few things tying Wayne Williams in solid physical terms to the murder victims. Everything else was circumstantial. But this fiber evidence has come under intense scrutiny. And there is some controversy about what its relevance really should have been. Because we have to ask ourselves how many other people had carpets or textiles which could produce similar looking fibers. That is very difficult to determine. And it wasn't a question really that was carefully asked at the time of his trial. So that's an example of the kind of controversy that can arise around fiber usage. There are ways of examining the fibers to look at to actually determine their color in scientific terms, rather than simply looking at them and making a judgment saying, I think this is reddish, or I think that this is reddish brown. And there are various ways of treating them with light and then looking at the results. Don't worry about this over much. The spectrophotometers, what spectrophotometer does is it examines the wavelengths of light, which are either produced by a substance for instance, if we shine ultraviolet light on it and fluoresces, or else if we put light through it and see what light gets absorbed, what light gets transmitted. All of these are measured by a spectrophotometer. So spectrophotometry may tell us a lot about the color and light properties of different fibers. But all of these things, as I say, they are really mostly provide supportive or corroborative evidence. It is nowadays very, very unlikely that evidence of this sort would be key to a conviction. This fiber evidence has become somewhat suspect, especially since the FBI studies that showed such a high error rate in analysis and interpretation. Nonetheless, the fibers at the scene of a crime are very carefully collected. Again, these may be minute. It requires very, very careful examination of a crime scene in order to find hairs and fibers which might be significant. And it may be extremely tedious. There may be a lot of them to collect. They're usually packaged very carefully in paper, in envelopes, paper, and then in paper bags if it's textile, things like that. And they have to be carefully collected so that they're not damaged. It's very easy to crush cross-sectional shape, for example. And in addition, the final really dangerous thing is that we carry fibers with us the whole time. For heaven's sake, we have heads full of hair. We have clothing made of fibers, so the investigators themselves may carry in fiber and contribute fibers. This is like Locard's principle, okay? Everybody who enters the scene leaves something and takes something away. Fibers are one of the things that we leave behind. For this reason, very often at a crime scene, investigators will dress in protective gear. They will cover their heads. They will cover their clothing. They will wear gloves. They will wear booties on their feet, etc., etc. And one of the reasons they do that is so that they do not contribute hair and fiber evidence and potentially DNA evidence to a crime scene. Okay, so that is it for today. And then I'll see you again on Wednesday. Remember, Wednesday is a live class. Very few people are coming to the live class. It would be nice to see some of you there. It is an opportunity for me to see you, but also for you to ask questions.